Welcome to today's session, sex linked inheritance, continuation, hemophilia. In the previous session yesterday, I have already given you information about hemophilia and the transmission of the gene for hemophilia from parents to offsprings. I have already given you one question to solve the flow of recessive gene from parents to offsprings. Here I will show you, I will help you to solve the same. Listen carefully here. This is the carrier female with small h gene for hemophilia. Hemophilic male, we can assume this as living. It possesses a single allele. There won't be any allele on its Y chromosome. As I have already told you, this condition is known as hemizygous condition, single allelic condition present in the male individual for X linked inheritance is known as hemizygous condition. Now, here the gametes produced are one with capital H allele and the other with small h allele. Here it is X chromosome with small h allele for hemophilia and Y chromosome without any allele for hemophilia, or even the normal allele for this character. You can see the random fusion of gametes and the possibilities among the progenies. Here in this condition, in this particular cross, there is the production of a female individual with small h allele. So, it is a carrier female. See this fusion of gametes. It is a male individual and it is normal. It is normal in the sense it possesses capital H gene for normal hemoglobin. Capital H gene. Then see this particular case. Here both the X chromosomes contains allele for hemophilia, small h allele. It is female individual and it is hemophilic female. Here comes the emphasis. A female individual can become hemophilic in only one particular condition. You can see here it is in homozygous condition. Both the X chromosome must contain the allele small h for hemophilia. Then only the female individual will be affected. That is the reason why hemophilia is very less among the females. Chance of occurring hemophilia is less among the female compared to males. Males are more prone to hemophilia because of this single allelic condition or even hemizygous condition. See, this particular random fusion leads to a hemophilic male individual. Now, you can see the second case, Haemophilia carrier female, that is heterozygous, one X chromosome with a normal allele and one with a hemophilic allele. This is a normal male with X and Y chromosome. See here, the possible progeny is female normal, and this particular X chromosome, this particular gamete, when it fuses with the Y chromosome, here there is the production of a hemophilic male. So, how this particular male individual becomes hemophilic, and from where this gene for hemophilia is transmitted? You just see, it is transmitted from its mother. So, from mother to son. Now, again this particular allele present in the X chromosome of the male individual will move to the female. So, from mother to son, then to daughter. So, this zigzag manner it is moving. So, this type of inheritance, that is called as criss-cross inheritance or zigzag inheritance. Here the same condition you can see here also in this diagram. Male normal and the female is carrier mother, and among the progenies the possibilities normal daughter, normal son, carrier daughter, and this is the affected son. So, from mother the allele moves to son, then from son it will move to his daughter. So, it moves in a zigzag manner, that means criss-cross inheritance. Now, at another example for sex linked inheritance, that is X linked inheritance. It is color blindness in man. Color blindness in man, it is known as red-green color blindness, and the gene for this is a mutant 1, dominant gene mutated to recessive 1, a mutant recessive allele present on the X chromosome is responsible for causing this disease. So, it is also just like that of hemophilia. It is also an example of sex linked inheritance, X linked inheritance and recessive mutation. The mode of transmission of this affected allele is again in a criss-cross pattern or in a zigzag manner, from female to male and from male to female. Here it is the inability to distinguish red color from green color. It is due to the lack of one primary cone pigment present in the retina. Retina contains rod cells and cone cells meant for vision, cone cells meant for color vision and rod cells meant for dark and light vision or black and white vision. So, the pigment present in the cone cells, that is iodopsin, that is missing. So, it is not able to distinguish red color from green color. So, it is known as red green color blindness. Males are usually more affected again due to single allele condition present in the X chromosome, hemizygous condition just like that of hemophilia. And females, the chance of occurring this particular disease is very less, because both the X chromosome must contain the recessive allele. That is, only in homozygous recessive condition they are producing this disease, color blindness. Heterozygotes are again the carriers. I have already told you it shows criss-cross pattern of inheritance from mother to son and son to daughter. I will show you the pattern of transmission of this particular allele for color blindness. You can see here normal female individual with a normal allele capital C in both the X chromosomes. This is the color blind man. The capital C gene mutated to small c gene. It is linked to the X chromosome. Now, the phenotype of the progeny is possible progeny is here capital C and small c alleles together. This will be a carrier female because it carries the small c allele for color blindness. Here this is a normal male individual, because the X chromosome contains capital C allele. Now, among the F2 generation, the possible phenotypes progenies, one female that is normal, one male that is also normal. This is color blind male individual. This is carrier female. So, these are the different types of progenies appearing in 1:1:1 ratio regarding male as well as female in this phenotype. Now, the next topic is sickle cell anemia. It is an autosome disorder, an autosomal disorder. The gene or the allele for this particular character is present in the autosome, and it is due to a recessive allele. The autosome linked recessive trait is sickle cell anemia. It is transmitted when both the parents are carriers of this particular gene. That means when both the parents are heterozygous, it is transferred to the next generation progeny. Progeny will be affected by sickle cell anemia. Controlled by a single pair of alleles, we can represent it as normal one as HBA and the mutated form that is HB small s, recessive allele for sickle cell anemia. Homozygous individuals, that means HBS, they are the affected individuals, they are having sickle cell anemia. It can be seen in both males as well as in females. because it is an autosomal linked character. Here also the heterozygotes are the carriers. They have HbA allele for normal hemoglobin and HbS allele for sickle cell hemoglobin. You can see the reason for this particular case here. What is the cause for sickle cell anemia? Why it happens? Here see, this is the normal gene, the part of the DNA that produces the polypeptide chain meant for normal hemoglobin. It possesses CAG and the corresponding sequence is CTC. It undergoes mutation. Here A is replaced by adenine nucleotide, that is replaced by thymine nucleotide T. Mutation happens only in a single particular nucleotide. Only on a particular point there happens the mutation. That means the adenine nucleotide is mutated to thymine nucleotide. A is replaced by T. Now, because of this particular mutation, this mutation happens only on a particular point, it is affecting only a single nucleotide. So, it is known as a point mutation. It is known as point mutation. So, because of this particular point mutation, this normal codon present in the mRNA that changes from GAG. You see, C will be replaced by G in the mRNA during transcription. Here you can see the complementary base pairing. In DNA it is ATCG pairing, but in RNA it is AUCG. So, C will pair with the G and T will be pairing with the A and C will be pairing with the G again. But here in the mutated one, you see C will pair with the G and A will pair with the U, because it is RNA and this is DNA, AU, CG pairing. C pairs with G. The normal, here the codon, this is the normal codon present in the mRNA with the GAG nucleotides. It is the codon mutated one. Here you can see A is replaced by U. GAG codon, where A is replaced by U. So, the glutamic acid, that is the normal amino acid, that is replaced by valine. So, this becomes the mutant protein and this is the normal protein for the production of normal hemoglobin. This mutant one, that is abnormal hemoglobin for the production of sickle cell RBC. Same I will show you here. You can see the other amino acids also in this case. HbA gene, that is the normal gene. CTC and the complementary pairing GAG. Here it is CAC and GTG. This is HBS gene, gene for sickle cell anemia. mRNA is GAG and here it is GUG. You see the different amino acids: valine, histine, leucine, threonine, proline, glutamic acid. Here also it is glutamic acid. Now, this is glutamic acid, you can see in the 6th position here. But here you see that glutamic acid is replaced by valine on the 6th position. So, this particular condition is due to point mutation, which affects a single nucleotide present in the gene as well as in the codon. This will contribute to normal polypeptide chain for normal hemoglobin, and this leads to the production of HBS peptide chain, which will produce abnormal hemoglobin and hence sickle cell RBCs. Now, you can see the shape of the RBCs here: normal RBC, biconcave and enucleated with normal hemoglobin. What is the advantage of enucleated RBCs? It is to carry maximum amount of hemoglobin. Here you can see the change in the shape of the RBC. It is sickle shaped RBCs due to abnormal hemoglobin formation. So, as I have already told you, in the 6th position of the polypeptide chain, normal hemoglobin, where glutamic acid is replaced by valine amino acid in the mutated one or affected individual. It is in the 6th position of beta globin chain of the hemoglobin molecule. Hemoglobin molecule, the globin chain contains alpha and beta, 2 alpha chain and 2 beta chains. So, in the beta chain on the 6th position, the glutamic acid is replaced by valine amino acid. That means again the codon is 6th codon, because one codon will code for one amino acid. In the 6th codon, in the beta globin gene, there is a single base substitution. I have already told you only one particular nitrogen base is affected, that is GAG, codon changes to GUG. So, here you can see in the place of A, here comes U. So, because of this nitrogen base or even nucleotide change, substitution, this happens. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension and changes its shape into sickle shaped. That result in the production of sickle shaped RBC. Even though it is an autosomal recessive disorder, in African countries there occurs some advantage. The areas where it is much more prone to malaria, This particular disease has an advantage. Heterozygotes with a capital H, B and A gene, similar, and HBS allele, they have some selective advantage over the normal individuals. It lowers the occurrence of malarial disease. Since the RBCs are having abnormal hemoglobin, the shape of the RBC becomes sickle shaped, they are deprived of oxygen, they undergo polymerization, and hence these parasites can't live inside the RBCs. So, they will die, and by that time they will the parasites will never get enough time for multiplication in the body, and body's own defense mechanism will kill the remaining parasites. So, the heterozygous sickle celled anemic individuals, sickle celled means here they are carriers, heterozygous individuals means they are carriers, they protect the person from malarial infection. So, that is a kind of selective advantage in malarial prone areas. Now, we are moving to yet another concept, an inborn error in metabolism, that is phenylketonuria. It is a metabolic disorder seen from the time of birth onwards. Here the reason for this is also a recessive mutant gene which is present in the autosome. So, it is caused due to autosomal recessive mutant allele. Due to the presence of this recessive mutant allele, there lacks the production of an enzyme. Name of the enzyme is phenylalanine hydroxylase. This particular enzyme, which is needed for the conversion of phenylalanine, an amino acid, into yet another amino acid, tyrosine. Since the normal gene, dominant gene is mutated into recessive one, the enzyme is not produced. Here there is a concept, one gene, one enzyme concept. One gene will produce one protein which modifies into an enzyme which is needed for the metabolic pathway, a specific metabolic pathway. Hence, this is a metabolic error. So, due to the lack of this dominant gene, normal gene, the enzyme is not produced. The enzyme is phenylalanine hydroxylase, which is needed for the conversion of phenylalanine into tyrosine. So, what will happen is phenylalanine is accumulated in the bloodstream of such individuals and converted into so many derivatives, phenyl, pyruvic acid as well as so many other derivatives. An accumulation of all these compounds in the brain that leads to mental retardation. Moreover, it will reach the kidney through bloodstream and it is given out through urine. So, when it is given out through urine and when the urine is exposed to air, it turns into black in color. So, this particular disease, phenylketonuria, is known as black urine disease. Now, we are moving to the next concept here, chromosomal disorders. You know, chromosomal disorders are caused due to either addition or deletion or inversion or translocation of chromosomes. Any change that happens to the chromosome that leads to a disorders which is known as chromosomal disorders. Here comes the basic concept ploidy. You have already heard about it: haploid, diploid, triploid, tetraploid and pentaploid and polyploid, like that. So, what is ploidy? Ploidy refers to the number of a set of chromosomes present in an individual. If an individual possesses only one set of chromosome, then it is haploid. If it possesses two sets, diploid. If it possesses many sets, more than two, then it is polyploid. So, polyploidy means addition of many sets of chromosomes or presence of many sets of chromosomes in an individual. Now, you might have heard about aneuploidy, addition or even deletion of an individual chromosome to the normal diploid set of chromosomes. For example, we human beings have 2n is equal to 46 chromosomes. Suppose the 21st autosome is added again, then we will become 47. 2n is equal to 47. We can represent it as 2n is equal to 46 plus 1, because one chromosome alone is added here in the normal diploid set. So, if similarly, if one chromosome is removed, deleted, then also the condition we will call it as aneuploidy. For example, 2n is equal to 46 in humans. Suppose one X chromosome is missing in the female individual, then such females, they will become 2n is equal to 45. So, 2n minus 1 condition. And in trisomy and in the case of Down syndrome, it is also an example of aneuploidy where there occurs addition to the normal diploid set. That we can represent it as 2n plus 1. If one chromosome is deleted, we will write it as 2n minus 1. So, both are the conditions of aneuploidy, where there is addition of an individual chromosome or deletion of an individual chromosome from the normal diploid set. Here comes the disorder for aneuploidy, for chromosomal disorder, that is Down syndrome. You know, Down syndrome is an autosomal disorder. In every individual there are two types of chromosomes, as I have already told you, autosome as well as sex chromosome. So, here it is an autosomal disorder. You know, the karyotype is chromosome complement of an individual. We can represent it as 2n is equal to 47. Normally human beings 2n is equal to 46. Here one chromosome is seen in triplicate. That means which chromosome? The 21st autosome. So, it is known as the 21st chromosome is represented 3 times in this particular affected individual. So, it is known as 21 trisomy. 21st chromosome, that is represented or seen 3 times in this particular individual. So, it is an example of aneuploidy. One chromosome, 21st autosome is normally it is seen only twice, but one more is added. So, it becomes 3 times. So, that condition is an aneuploidy. It was discovered by Langdon Down, so known as Down syndrome, or common in Mongolians, so known as Mongolism. You know, late marriage and late pregnancy above the age of 40 in women, that most of the time leads to abnormalities in the fetus, and they are likely to produce such children with Down syndrome. So, it can be rectified, it can be detected, not rectified, it can be detected by prenatal diagnostic methods, amniocentesis, and such pregnancies can be eliminated. It is due to the non-disjunction of homologous chromosomes, that is the 21st autosome. You know, in a normal individual, 21st autosome is present only 2 times, and that pair of chromosome, you see here, I am representing here as this as the 21st homologous chromosome, this is the autosome. One maternal chromosome and the other paternal chromosome. Now, during gamete formation, normally it must move to 2 different, segregate and must move to 2 different gametes. But here what happens is, these 2 chromosomes, they are moving to one particular gamete itself. So, the other gamete is without 21st chromosome. Now, this type of one egg, when fuses with the normal sperm with 23, n equals 23 chromosomes, here it is n equals 23 plus 1, it leads to the production of 2n is equal to 46 plus 1, that is 2n equals 47 chromosomes. So, this condition, normal during gamete formation, the members of the homologous chromosomes must segregate and must move to 2 different gametes. But here it is not segregating. Hence, we are using the term non-disjunction. You see, junction means union, to be remain together. Disjunction means separation. Non-disjunction means non-separation. So, due to the non-separation of these homologous chromosomes, that is the 21st autosome. This condition occurs. In this case, a few victims of this Down syndrome, they grow up to maturity and they can produce babies, both Mongolian, that means babies with the Down syndrome, and normal children at equal proportion. So, there is a chance for getting a baby with the Down syndrome as well as a normal child. Such individuals having Down syndrome are more prone to some other disorders. One is leukemia, the other is Alzheimer's disease. Here you can see some examples, some pictures of these affected individuals. People with the Down syndrome. Now, coming to the symptoms of this particular disease. What are the different symptoms? One is short stature. The other one is small, round, moon-like face and head. Furrowed tongue. They have a broad and flat palm with palm crease. Mentally, physically and psychologically. Physical, mental, psychomotor development is retarded. They have round face, broad forehead, congenital heart diseases, and many lobes are there on the tip of the fingers. Short neck, stubby fingers, underdeveloped gonads, exogens, genitalia, a Mongolian type of eyelid fold. They have flat nasal bridge, so flat nose, permanently opened mouth, projecting lower lips and protruding tongue. So, all these are the different symptoms that we can see in a child having this particular Down syndrome. Now, coming to sex chromosomal abnormalities. Here you can see Klinefelter syndrome. It is a male individual showing most of the characters of female individual. So, male showing female characters, that is Klinefelter syndrome. It is due to sex chromosome abnormality disorder. Why it happens? An addition of an extra X chromosome with the normal XY chromosome. So, the karyotype of this particular individual, that is XXY. Again 2n is equal to 46 plus 1, 47. One X chromosome is added here with the normal diploid content, chromosome content. So, it is XXY 2n equals 47. 7. Again it is an example of aneuploidy, where we can see one X chromosome is added. So again the reason is non-disjunction of X chromosomes seen in the female individuals. So I will show you the case here. Two N is equal to 46 chromosomes, that is 22 autosomes plus 1 pair XX chromosome in the case of female. During gamete formation, (22) autosome plus 1 X chromosome in one of the egg, and in the other one also it must be the same, 22 autosome plus X chromosome. This is the normal gametes produced by the separation of homologous pair of chromosomes during gamete formation. Now here in this case, if these two X chromosomes, they are not segregating like this, then there is the production of 22 autosome plus XX chromosome in one particular egg. Here what you can see here is both the X chromosomes are moving together, they are not separating, they are moving together into a single gamete. If such an individual, such a gamete fuses with a normal sperm with a 22 autosome and XY with a Y chromosome, then this fusion leads to the production of 44 autosomes plus X, another X and this Y chromosome. So this particular condition, here the individual possesses 2N is equal to XXY karyotype, that is equal to 47 chromosomes. So this particular condition, the condition in which the homologous chromosomes, they are moving together into one gamete, this is known as non-disjunction of chromosomes, homologous chromosomes. So here also in Klinefelter syndrome, the reason for this particular syndrome is non-disjunction of X chromosomes present in the female individuals at the time of gamete formation. So it results in the production of an egg with both the X chromosomes and the other one without any X chromosome. Now what are the symptoms of this particular disease? Such individuals are sterile, they are not able to reproduce, they are having poorly developed testicles, they show female characters, for example, breast development, normal character of female, it is seen in this male individual, and this condition is known as gynecomastia. They are abnormally tall in stature, have long legs, obesity, sparse body hairs, again at another female character. Now coming to yet another syndrome, sex chromosomal abnormality disorder, Turner's syndrome. Here the female individual is showing male characters. In Klinefelter syndrome, male is showing female character. Here female individual is showing some of the male characters. It is caused due to an absence of one X chromosome. Normally females have a XX chromosomes. Here one X chromosome is missing in this individual. So female will show the characters of males. Here 2n is equal to 45 chromosomes. One X chromosome is missing from the normal chromosome complement of the female individual. So it is 2n is equal to 45. That means X 0. One X chromosome is missing. Again it is an example of aneuploidy. Here deletion of one X chromosome takes place from the normal diploid set. Symptoms, they are also sexually sterile individuals. They have rudimentary ovaries, sex organs in females, not developed. Short stature. In Klinefelter syndrome, tall in stature. Here it is short stature. The skin in the neck region is just like a webbed neck. They have a broad chest, just like males. Reason is, it is due to the production of an abnormal egg with autosomes only. There won't be any sex chromosome. So such a type of gamete, when fuses with the normal sperm having X chromosome or with a normal egg, when fuses with abnormal sperm, that also result in this condition, X0 condition. They have underdeveloped breast. The feminine character breast development not normal, reduced ovaries, small sized uterus not well developed, short stature and many other characters, male characters, heavy neck muscles. They have a narrow hips, normally ladies in females, wide hips or widening of the hip region. They does not menstruate or ovulate. So here you can see the karyotype chromosome complement of this particular individuals. These are the different autosomes you can see here, and this is the sex chromosome complement with only one X chromosome. No, the second X chromosome is missing in this individual.